Good morning, and welcome to Psychiatry Grand Rounds. This morning I have the pleasure of introducing Scott Holbrook. He is the Vice President of Research and Development for Pharmacologic Holdings Incorporated. He has over 20 years experience in the field of molecular imaging and therapeutics, which encompasses all phases of radiopharmaceutical drug development, including target identification, GMP manufacturing, clinical trials, translation, and post-approval manufacturing. After completion of his Bachelor's of Science degree in Nuclear Medicine Technology from Wheeling Jesuit University, with clinical rotations in Pittsburgh and University of Pittsburgh Medical Center, Mr. Holbrook worked for PETNet Solutions in Knoxville, Tennessee. His areas of focus include new cyclotron program development in the southeastern United States at facilities which include the University of Tennessee, Vanderbilt University, University of Arkansas, and University of Louisville. After completing an initial public offering, Mr. Holbrook left PETNet Solutions and co-founded 7 companies offering a spectrum of healthcare technologies, including drug development, GMP contract manufacturing, pharmaceutical services, and molecular imaging. While completing a Master's of Science degree in molecular imaging and therapeutics from Regis College in Weston, Massachusetts, Mr. Holbrook assumed the role of Vice President of Research and Development for Pharmalogic Holdings Incorporated at Boca Raton, Florida. His roles include oversight of collaboration with pharmaceutical companies, researchers, healthcare organizations, and universities. Mr. Holbrook participates in strategic decisions regarding pharmaceutical development, regulatory matters, and translation of new drugs into the clinical setting. During his career in molecular imaging, Mr. Holbrook has served on more than 50 internationally recognized committees and boards of directors established by international medical societies and government agencies. He has served as president of the Society of Nuclear Medicine and Molecular Imaging on 2 occasions and on the board of directors for 5 years. During his tenure, he represented the SNMMI at key meetings with various government and regulatory bodies, including the FDA, CMS, U.S. Congress, Joint Commission, and Nuclear Regulatory Commission, as well as the Conference of Radiation Control Program Directors and various credentialing bodies. Mr. Holbrook chaired the SNMMI Task Force for Radiopharmaceutical Compounding and was a key contributor to the Drug Quality and Security Act passed by Congress in 2013 and subsequent FDA guidance documents. Mr. Holbrook has served as a reviewer and associate editor of the Journal of Nuclear Medicine Technology. He has authored over 100 papers, presentations, and book chapters, including the Society of Nuclear Medicine's Guideline for PET Tumor Imaging, Procedure Guideline for Tumor Imaging with FDG PET-CT, and the Joint SNM and European Association of Nuclear Medicine Guideline for Tumor Imaging, FDG PET-CT. EANM Guidelines for Tumor Imaging. He has organized and presented at numerous national and international conferences. Please join me in welcoming Scott Holbrook.
Sorry about that. I should have just sent you a couple bullet points for that. But thank you for inviting me to give today's lecture. I was just saying earlier, it's great to be here in Tennessee. I actually live in Johnson City, and I don't get to spend as much time here talking about the things that we do as I would like. So travel around quite a bit. And the field that we're going to talk about today is evolving quite rapidly and grown quite a bit in the last 10 years. When I originally trained in Pittsburgh in the late 90s, 96, the drugs that we're discussing, there were really no FDA, not only were there no FDA approved drugs really in this area, but there was no pathway even established by the FDA for approval of these drugs. And the technology that I'm going to speak about was being invented at that time. And fortunately, I'm originally from this area, but training in Pittsburgh, I was able to train with researchers who were working in these areas that invented a couple of really important technologies in the time that I was there, one of those being C11PIB, which is a drug we're going to talk about a little bit today, and the other is the instrumentation which we use to image patients for both research, clinical trials, and routine clinical use as well. David Townsend was in Pittsburgh at the time and is one of my mentors, and he invented PET-CT in the time that I was there in Pittsburgh. And it's really exciting. When I left Pittsburgh, the PET-CT gained FDA approval, and he was awarded Time Magazine Medical Invention of the Year and was supposed to be on the cover of Time magazine and was bumped. He was still in the magazine as Medical Invention of the Year, but was bumped from the cover to further back in that magazine because on the cover of that particular point in time was the election recount between Bush and Gore that was going on. So he got bumped, unfortunately, but it's still pretty exciting nonetheless. So, but originally getting into this field, the focus of the field was purely a diagnostic tool that was just meant to aid clinicians in primarily diagnosing patients with cancer and kind of evaluating the location of their disease or their response to therapy, just using basically one clinical drug at that time, which was fluorodeoxyglucose, which we'll talk about a little bit. But in the last 5 to 6 years, some really important changes have occurred in our field based on our unique ability to label biomarkers, image their biodistribution, quantify the presence of disease previously undetected, and the role of these biomarkers in serving as surrogate markers for future therapeutic drugs. And kind of this, the field of personalized medicine, companion diagnostics, and theranostics has evolved quite rapidly in the last 5 to 7 years. And my role has translated quite rapidly from someone who was just focused on basic provision of diagnostic drugs for clinical management of patients to someone who now most of my work is working in collaboration with researchers and drug companies who are certainly interested in downstream applications of these drugs in a clinical setting for diagnostic purposes, but are even more interested in the ability to use these biomarkers to differentiate patients who would benefit from one therapeutic drug versus another, to identify targets for therapeutic intervention. And so some really exciting new therapies that are being approved rely heavily now on this technology, which is very new, rapidly evolving, regulatory pathways only recently approved. And so I would say that even major institutions that have been major players in research and development of new technologies to improve patient care in the last decade are far behind areas even like Johnson City in being able to provide these drugs because of the lag of regulatory expertise and inability to provide these drugs in an FDA-approved environment. So I spend a lot of my time working with academic institutions and pharma companies who are interested in this technology for development of their therapeutic drugs. So just the first 3 slides, the kind of obligatory comments would be, I do have financial interests in that. I do work with pharma companies now who are interested in developing therapeutic analogs of the biomarkers that we manufacture. I'm also on the board of directors and a member of senior management of a pharma company that manufactures these biomarkers. And so my current disclosures are primarily I work with GE Life Sciences as the pharma company. And they subsequently work with other pharma companies who are interested in using these novel biomarkers to develop therapeutic agents. And pretty much any pharma company that you could think of is collaborating currently with GE Life Sciences in their portfolio of biomarkers. And then the name of my company is PharmaLogic Holdings. And also I will be discussing drugs that are in early to mid-phase development, also a couple of drugs that are clinically approved, but I'll be sure to let you know which ones are still investigational and which ones are improved as we move through the talk. So my objective today is to give you a basic understanding of what these biomarkers are and why they're important, how they're manufactured, major obstacles and challenges associated with providing these biomarkers for researchers or clinicians in any given region, how they are now very useful companion diagnostics, theranostic agents, and their importance in development and drug discovery. And then, and again, we'll kind of expand on that, and I'll give some examples, current exciting examples of how these biomarkers are being used currently, even here in Johnson City, in drug development and clinical trials for therapeutic agents. So some of the unique challenges of these drugs. So each biomarker that we manufacture essentially has 2 components. One component is a ligand, which will, it's designed to target a specific receptor or molecule or something unique about a disease process that we want to study. The other component of each biomarker that we manufacture is a radionuclide, which produces a signal. So the kind of overall concept is that the ligand will target the disease. It'll also carry the radioactive molecule, which we create at our lab, and produce a signal so it's detectable and quantifiable. And so kind of the first step in that process is that we actually have to create a radionuclide for the labeling of these ligands. And these radionuclides have short radioactive half-lives, so they have to be produced in relatively close proximity to the area of use. Some of the drugs that we manufacture have a radioactive half-life component labeled with fluorine, which has a half-life of 108 minutes. Others have radioactive half-lives as low as 10 minutes or 2 minutes. So you can see that the radius of use of these drugs can be very limited to where they're produced. And so essentially, the first step in the process is we accelerate ions inside this circular instrument called a cyclotron until they reach kind of a maximum energy, and we allow them to escape their orbit and bombard a non-radioactive target material, making it radioactive. So in the case of fluorine-18, we bombard non-radioactive enriched O18 water, and these particles strike the nucleus of non-radioactive O18 water. A proton replaces a neutron, and now you have radioactive fluorine-18. It's as simple as that. Some of the different radionuclides that we produce in the cyclotron, O15, again, a half-life of 2.1 minutes, carbon-11, and we don't really use O15. It's used on a very limited basis around the US and Europe. Carbon-11 has a half-life of 20.4 minutes, N13 ammonia, which we make very routinely 10 minutes, and fluorine-18, 108 minutes. The nice things about these drugs is that you notice that they are essentially similar to or analogs of the basic building blocks of life. So they're very discrete once we couple them with a ligand, and they're able to actually provide nice surrogates for any biochemical process we want to study. For example, if we wanted to study perfusion, oxygen-15 is very useful in that it behaves as oxygen that your body would use for glycolysis or Krebs cycle. Carbon-11 obviously recognized by the body as carbon, and we can label lots of compounds with carbon, follow the tracers, they emit the signal, and we can detect different forms of disease or biological processes with these carbon-11 labeled molecules. So those are the radionuclides, and then the actual ligands themselves, you can see this kind of, this is a very busy slide and not to go into all of these areas, but you can see these are kind of the different areas that we've been approached by researchers and drug companies who have interest in studying diseases. You can see cardiovascular disease, neurodegenerative diseases, neuropsychiatric diseases, oncology is a big area for us. And so we are currently in the process of developing with researchers and pharmaceutical companies ligands that will target diseases in each of these areas and serve as companion diagnostics to aid in development of therapeutics that they may be working on, or diseases even earlier phases that they want to study a specific scientific principle. So once we have the radionuclide produced in the cyclotron, the ligand is actually labeled in a different area of our lab. And you can see here kind of the environment is, it's an FDA environment. It's a clean room environment. It has to meet very stringent FDA requirements. You can see all of the agencies that we're regulated by. And again, we talk about the FDA a lot, but because we're dealing with radioactive materials that we want to inject into human patients or human subjects or even animal subjects, you can see we're also regulated by the Nuclear Regulatory Commission, the state departments of radiologic health. In addition to the FDA, we're regulated by the Board of Pharmacy. When we work with institutions like ETSU, of course, our drugs are reviewed by the IRB, or in the case of very small biodistribution safety studies, if there's an RDRC, they may look at our drugs in what's called an IND, a packet, before approval of the use of the drug in a human subject. And then, of course, pharma. Even though pharma owns the intellectual property for all the ligands that, or a good bit of it, they cannot manufacture these drugs for their studies because of the short radioactive half-life. So pharma will always have to rely on local or regional manufacturing locations to provide these molecules for researchers, academic institutions, and for support of multi-center clinical trials. And so we kind of operate under the traditional IND for research drug pathway, new drug applications for pharma drugs that have made it through clinical trials and are approved, and then abbreviated NDA for the generic drugs that we manufacture. So very stringent quality obstacles that we have to deal with to meet the quality specifications of all these regulatory bodies, as well as just the physical characteristics of the drugs that we make. They're difficult to manufacture and have very short radioactive half-life, so it's a very unforgiving environment to prepare these drugs. And then once the drug is prepared and shipped off from the laboratory to an institution where a clinical trial may be going on, or a clinical patient actually needs these drugs, then the data is actually acquired on one of these units, and these are readily available throughout the entire area. There's one across the street, they're in Kingsport, in different locations locally. And so what happens is the tracer is administered to the patient and given sufficient time to reach its target, and a signal is emitted from the patient, which is detected by these instruments and turned into data. And you can kind of see, this would be an example of a signal coming from the deep inside a patient's brain. The data leaves the patient, and the signal is detected by the scanner and turned into an image. And what's really important about the data is we're not just visually looking at an image set and having, for example, a radiologist make a report about what they see on the image. What's really important about PET data is it's highly quantitative. And so the data is very useful for researchers and pharma companies who want to quantitate the kinetics of this drug and its accumulation in a target over a period of time. And also we're able to quantify the response of a given pathology to a challenge or to a drug, for example, that also targets the same target. So this is one of the drugs that I mentioned earlier, fluorodeoxyglucose, that we make that's commonly used for clinical patients for cancer evaluation, so diagnosis, staging, restaging, and you can see the data. This drug is actually a glucose analog, and so it's a great tool for imaging cancer patients because most cancers that we image will overexpress glucose transporter proteins. They have kind of unchecked an upregulation of glucose metabolism, so we inject this radio-labeled version of glucose, and the cancer cells, when they encounter the glucose, they can't differentiate between this glucose and dietary glucose. It's transported intracellularly, kind of disproportionately in cancer cells compared to normal tissue, and then the patient is imaged, and we detect the signal, and we're able to quantify the absence or presence of abnormal cancer tissues, and we can actually quantify the amount of drug that's concentrated in the tissue. So this is just kind of a basic routine clinical use that you would encounter on a daily basis at any area medical institution. Another example of a tracer that we make in 13-ammonia, this is used for non-invasive quantification of coronary artery disease. This is a tracer that has a 10-minute radioactive half-life, and something that we actually developed here, a new method for in Tennessee, and we're actually the leading institution for this drug with our method here locally, so it's something we're proud of. And so that's just a kind of an example of some of the clinical uses, but the really exciting area that we're here to talk about today and what we work on with pharma and researchers is the use of novel tracers and companion diagnostics and drug development and discovery. And so again, FDG, the drug that I just showed you, the oncology patient, is already approved for use in differentiating patients with Alzheimer's dementia from frontotemporal lobe disease, for example. It's been approved for quite some while by Medicare, and you can see the marked difference in the frontotemporal lobe hypometabolism in this dementia patient versus the frontotemporal lobe hypometabolism in the patients with Pick's disease. This is a great tool, but unfortunately it's not particularly helpful for drug companies because these patients, the goal of the drug companies that we work with now is to kind of detect the disease at a very early state and test disease-modifying therapeutics. And so these patients, you could give them a drug that would target the amyloid protein or the tau protein in their brains, and you could remove that protein, but you wouldn't really expect to see any improvement in cognition for patients with disease that's this advanced. So we need to have biomarkers that are more sensitive and detect the disease differently than FDG at an earlier phase as to be used as a useful companion diagnostic. And so just to kind of give you an example of what's being done, the next couple of areas are in the psychiatric area. This is not my particular area of expertise. I had a new colleague join our company about a year ago who was the head of research at Columbia. And so this is actually work that she's very familiar with, but she donated a couple of slides to me. And so this is a drug called C11PHNO, which binds to D3 receptors. And so researchers who are interested in evaluating the presence and concentration of these receptors, and then potentially a challenge for the subject, exposing the patient to a challenge or a test, or even a drug to determine kind of the impact of that drug on the accumulation of the drug in the D3 receptors. So what we're actually looking at is competitive binding. And you can see basically the three panels of I hope I don't cut something off here. You can see these three panels are actually examples of three different drugs that are being used as a challenge, and you can see the competitive binding for these drugs and the D3 receptors. The D3 receptor, the goal is the tracer is administered, and then the drug has a higher affinity for the receptors than the imaging agent. And so it's kind of a measurement of affinity or action of the drug on these receptors by subtraction. And we don't currently prepare this drug at the lab here in Gray. Carbon-11 products, we do have capability to make carbon-11, but that radioactive half-life is particularly short. It's 20.2 minutes. And the other issue is that the specific activity for this drug has to be very high, and by that I mean the radio-labeled drug versus the non-radio-labeled cold product also compete for binding in these receptors. So even if you do successfully manufacture this drug, it has to have a very high specific activity, so you don't unintentionally label the D3 receptors with non-radioactive drug, because we can't measure that. So we don't currently manufacture this drug. This was done at Columbia University. And then here's another example of an F18 labeled drug called falipride, which labels the D2 receptors, and kind of the same idea here is we're looking for a competitive binding and the impact of a challenge or a drug on the D2 receptors and displacing the concentration of the radio tracer. So, and you know, even prior to the last two slides that I showed you that were human subjects, drug companies and universities alike use these small animal imaging devices called MicroPET scanners, for example. And preclinical work is done typically prior to administering these drugs to a human for safety, biodistribution, pharmacology, toxicology studies. So researchers will obtain mice or rodents that express a given disease, and they will go through a library of probes and assess the affinity for the probe of the target, assess the biodistribution of the probe. For example, if you wanted to target a CNS pathology and the probe wouldn't cross the blood-brain barrier, that would be good to know before you make it into human subjects. So we don't have access to this technology here locally in Johnson City. I typically work with researchers at the University of Tennessee in Knoxville who do the preclinical imaging work there. They have a preclinical imaging lab there. Most of the work that we do here, obviously, is for human subjects. And so kind of our area of work, not so much in neuropsychiatry currently, but in neurocognitive areas. And a lot of the recent activity has been surrounding targeting plaques that are associated with Alzheimer's dementia. So as you know, two types of proteins are unique to Alzheimer's patients versus other forms of dementia and must be present for the diagnosis to be confirmed. And these are the neurofibrillary tangles and the amyloid plaques. Both of those proteins must be present. And kind of what we see here is a drug that isn't really being used for research or clinical purposes. This was developed by Jorge Barrio at UCLA. And this drug labels both the tau proteins as well as the beta amyloid proteins in this patient. And so the basis for all of this work in neurocognitive research using PET biomarkers and companion diagnostics actually kind of began in Pittsburgh and was based off the original work that was done with a compound called Pittsburgh Compound B, or C11PIB. And so this drug was actually developed by imaging group, radiochemists, and pathologists. And they actually worked with the stains that were used to label these proteins at postmortem autopsy, and developed a radio-labeled analog of one of those stains, which became C11 Pittsburgh Compound B. And it labels the amyloid proteins, which are one of the two proteins found in the brains of dementia patients, which is a target for therapy. And you can see kind of across the top row of patients here, you see very little beta amyloid protein accumulation. That's the healthy control. And then across the bottom, you see a patient with Alzheimer's dementia, which has tons of this beta amyloid protein that we're labeling. So kind of the idea or the goal of this drug originally for research is that we are able to detect in vivo the presence of this drug. You know, waiting for patients to die and doing postmortem autopsy, that's kind of a tough endpoint to wait for your subjects to die. It would be nice to hopefully begin to show the benefit of disease-modifying therapeutics in living subjects. So, so this work was done several years ago in Pittsburgh, and several studies have been done to actually kind of show the correlation of these beta amyloid proteins in the brains of dementia patients. And so here's something that was published in Neurology in the late 2009, where a study was done with C11PIB that showed in these subjects that 55% of these subjects that have mild cognitive impairment showed increased C11PIB uptake, and 82% of those patients converted to Alzheimer's dementia within 3 years. And the other really interesting point that we have to hang our hat on in the absence of disease-modifying therapeutics is that only 7% of the PIB minus MCI cases converted to Alzheimer's dementia. So even though we don't currently have access to a disease-modifying therapeutic, which we're supporting those studies with these biomarkers, the benefit of having a PIB negative or an amyloid negative scan is that if you have a patient that presents with kind of a mixed clinical presentation, if they have a negative amyloid scan, then you can probably exclude them from kind of the Alzheimer's category of patients and look at another potential disease to treat. And so, so one of the issues with C11PIB is its short radioactive half-life of 20 minutes. And pharma companies that have an interest in using this as a companion diagnostic, gaining access to this molecule is very difficult. And being able to rely on it post-approval of some sort of disease-modifying therapeutic or even clinically ahead of that to rule out patients with Alzheimer's, having a molecule labeled with carbon-11 is not a very attractive option because you can only provide that drug locally at a very few number of institutions. So, so now there are actually 3 FDA approved. These are the FDA approved, and that says status phase 3. That probably needs to be updated, but this is actually an approved molecule now, and it's F18 labeled based off of the Pittsburgh compound B backbone, labeled with fluorine-18. It's approved by the FDA for use in patients with suspected Alzheimer's dementia. This one is owned by GE Healthcare. There's another one that was developed by a company called Avid, which was acquired by Eli Lilly. And this was the first one that was acquired, and so across the bottom row of this slide, you actually see this would be a healthy control. Across the top would be a patient with Alzheimer's dementia. And what's interesting about this slide is we actually participated in this study here locally. We were one of, I think, 21 sites around the country that supported the approval of this drug, and it was the first one approved in the class. And so kind of the test of truth for this study is we enrolled subjects who were terminally ill, either with dementia or some other disease, performed the imaging study, and then harvested their brains and conducted postmortem autopsy and was able to prove correlation between postmortem autopsy and the findings of this imaging study. So that was what led to the approval of this class of agents, and subsequently the other companies that own approved agents in this class had to repeat a similar study. So GE, Eli Lilly, and then an Indian company called Pier Mall, which owns this drug, also completed a similar study. And so you can kind of see across this spectrum, you see frontotemporal lobe, vascular dementia, and the healthy control. You don't really see a lot of increased amyloid burden in those patients. But so the area that we do see the increased amyloid burden is in patients with mild cognitive impairment, Alzheimer's dementia, but also dementia with Lewy bodies. So the positive findings with the study doesn't give you a definitive diagnosis of Alzheimer's dementia. Some of those patients with MCI will exhibit increased amyloid burden but may never convert to full-blown Alzheimer's. And also we have patients with dementia with Lewy bodies that can also have positive findings with these scans. But you can see the other pathologies, if they have a negative amyloid scan, it's safe to say that they don't have Alzheimer's dementia. And then lastly, this drug is an investigational drug. It was originally owned by AstraZeneca, and unlike the previous drugs that I showed you, there is no accumulation of this radio tracer. It has very high binding, very high binding efficiency for the amyloid protein. So there's no binding to any extra amyloid protein in this patient's brain. So a lot of excitement around these trials and approval of these drugs a few years ago, and even ahead of the FDA approval, what was really interesting is that the Alzheimer's Association, for the first time in over 2 decades, nearly 3 decades, kind of changed their classifications of Alzheimer's dementia clinical diagnosis. And they added a new category called Preclinical Alzheimer's disease. And this is based on brain biomarkers, so either biomarker data obtained from these imaging studies or cerebral spinal fluid confirmation of these proteins were the 2 ways that you could categorize a patient into this class. And so this was really important recognition by the Alzheimer's Association, and also really important for companies who aim to create disease-modifying therapeutics for these drugs, that we have now this new category of patients and subjects that are being identified. And how do we enrich our clinical trials and target these patients? And the answer to that is that we use these beta amyloid protein labeling agents now for clinical trials in the disease-modifying therapeutic area to select patients prospectively for these diseases based on the presence of amyloid. So my medical society at the time of this was concerned about the potential for abuse of this drug and this technology, and was concerned about the integrity of data. And so we worked with the Alzheimer's Association to develop appropriate use criteria for the use of this agent clinically. So kind of the 3 areas that were identified were patients with persistent or progressive unexplained mild cognitive impairment, patients who satisfy the core clinical criteria for AD, but they have a mixed clinical presentation, so they could have some AD symptoms, but also symptoms of some other neuropathologies as well, and so their diagnosis remains kind of unclear and difficult. And then patients with progressive dementia at an atypically early age, so that would be younger than 65 years of age. And then the appropriate use criteria goes on to identify 7 other areas where it would be kind of inappropriate to use these drugs. So patients who kind of meet the core clinical criteria for probable AD, and they kind of fit nicely into that diagnosis already. To quantify the dementia, severity of dementia, so quantifying that concentration of amyloid protein to kind of determine the threshold or severity or stage of the patient's dementia. There have been a lot of studies done to show that there isn't a direct correlation between the quantification of that beta amyloid protein and the severity of the patient's dementia. Patients with mild cognitive impairment that's unconfirmed on a clinical exam, in lieu of genotyping patients with suspected autosomal mutations, the APO4 gene carriers. Just screening in asymptomatic individuals would be an inappropriate use, and then non-medical use for legal reasons or insurance reasons, et cetera, have been identified as kind of inappropriate in our AUC. So this is in place. This has been in place for a few years now. So these drugs were approved, and they were approved to meet, the clinical trials were designed to meet kind of the scientific question, which was, does the presence of beta amyloid protein in the images that we acquire match postmortem autopsy findings? And the answer to that was yes. So all 3 of these drugs in this class received FDA approval. But the problem is kind of the ethical question, and the argument amongst regulators and CMS and other groups has been, so, what is the potential benefit of that to subjects or patients, that we can quantify the presence of this beta amyloid protein? So CMS didn't grant permanent coverage for this to be routinely used for clinical use, but rather what CMS elected to do was to cover payment for these procedures in the confines of what's called a CED, or Coverage with Evidence Determination program. And so the goal of the CED was to make the molecule available for clinicians and patients, but in return for that, CMS wanted to gather outcomes data about the benefit of this molecule in clinical patient populations absent a disease-modifying therapeutic. So a study was created called IDEAS study, which stands for Imaging Dementia Evidence for Amyloid Scanning. And essentially there's 2 aims in this study. The first aim was to assess the impact of amyloid PET on the management of patients meeting the AUC that I just described to you. And the second aim was to assess the impact of amyloid PET on hospital admissions, ER visits, sort of all of these other things that impact patients with one of these neurocognitive diseases. So AIM-1 has been completed, and the results of that were just announced in July of this year. And so the AIM-1 results were actually quite good, and we found through this study that PET scans have changed the management of 66% of patients in AIM-1. And AIM-2, I think, is just beginning as we speak. And this is also a study that I'm proud to say that we're participating in here locally. I think the only location in the state of Tennessee is here at the Quillen Medical School in Johnson City. So we're actively recruiting and enrolling patients here. Dr. Goodkin and Dr. Kendall's group, and then my company is actually, we're manufacturing the biomarker for this study. So we're trying to show that there is a benefit in outcomes for these patients, even absent a disease-modifying therapeutic. I would think once a disease-modifying therapy has been identified and shown to be efficacious, then obviously these drugs would be a no-brainer, pardon the pun, to make these drugs available more routinely on a clinical basis. So the other important area, the disease-modifying therapeutic targeting drug development, this is another study. It was also supported here at the Quillen College of Medicine called bapineuzumab. It was a drug owned by Pfizer, and monoclonal antibody targeting these beta amyloid proteins in the brains of suspected Alzheimer's patients. And patients were selected for this study based on clinical criteria. Now, unfortunately, we all know that using established clinical criteria, that there is a margin of error, maybe as high as 20% in patients that are selected and thought to have Alzheimer's may actually have another pathology altogether. So using that tool to select patients for this study was prone to error from the get-go. And so this drug failed in clinical trials, obviously showing efficacy for a drug targeting beta amyloid in patients that have symptoms but don't have dementia was a difficult task. And most of the patients in this study also had more moderate disease. So removing beta amyloid protein from that patient cohort group and showing an improvement in cognition was probably also pretty lofty aim as well. But what was really interesting about this study and what we learned from it was that there was actually a small cohort of patients that had very early mild disease, and with the removal of beta amyloid proteins in that patient subgroup, there was actually an improvement shown in that patient. So drug companies got smart from the failure of this study, and then subsequent future studies, they kind of adopted what they learned and changed. And so kind of the theory now, the amyloid theory, is that with this beta amyloid protein detection in vivo in living patients with these biomarkers kind of can predate kind of this MCI period as much as 10 years. And this graph kind of shows when the data from these various methods, such as the clinical cognition test, clinical function, other imaging modalities as well, such as MRI or physical imaging modalities such as CT, kind of the changes occur kind of later on in the disease process. So theoretically, if one were developing a disease-modifying therapeutic for Alzheimer's dementia, we want to identify subjects as early as possible in the progression of their disease, put them on a monoclonal antibody therapy or other disease-modifying platform, target those amyloid proteins, and see if we target them early enough prior to the loss of healthy neurons, if we can make a change or improvement or delay the onset of dementia. So this was actually Biogen's study that was announced about a year or so ago. And what was interesting about this is this study is the first time that a change in amyloid protein accumulation was detected in a patient in one of these amyloid therapies. So this was actually a phase 1 study where I think 5 different cohorts of patients, one group was a placebo group, and then 4 groups with varying concentrations of drug were given the drug and followed with amyloid tracer imaging. And we can see after 1 year of treatment, no change in the placebo group, and the very, very high group we were able to show a marked decrease in amyloid protein in those patients' brains. And so the other interesting component of this trial is that patients are selected for this clinical trial and other similar disease-modifying therapeutic studies based on first having a positive amyloid scan. So patients would be referred for all of these studies based on clinical findings. They would have an amyloid scan. If the amyloid scan is positive, then we know it's probably a good idea to enroll them in an amyloid targeting treatment. If the amyloid scan were negative and the patient didn't exhibit a high degree of amyloid burden, then giving them an amyloid targeting therapy was probably not going to show any results. So there are several drug companies with studies in this area, Merck, AstraZeneca, Biogen, Lilly, Pfizer, and several others, and they're all using kind of this companion diagnostic model where patients are being selected into the study based on the presence of these beta-amyloid proteins. And so kind of some future directions are the slide that we discussed earlier, which showed the presence of higher amyloid burden in the brains of dementia with Lewy body patients and mild cognitive impairment, again, doesn't rule in Alzheimer's, because you could still have a positive amyloid scan and remain MCI or have dementia with Lewy bodies. So the other protein is beginning now to also be a focus in our area, and that's the tau protein. And so here's an example of an investigational agent, which is, again, labeled with F18. And you can see kind of the healthy control group here doesn't really exhibit any increased tau, even though you do see some amyloid burden there in that patient. That's kind of a normal background activity. But you can see that tau also, in an AD patient, there's a great deal of tau burden. So in theory now, with this drug and the approved amyloid drugs, we would be able to perform sort of an in vivo biopsy in these patients and confirm the presence of both tau and amyloid proteins. So we can become even more specific and better in our selection of these patients. And so then kind of the last slide I'll show you is an area that we're beginning to work in at the lab in gray. We're just qualifying to manufacture this drug. It's a drug called GE180, which binds to activated microglia, which are responsible for inflammation, neuroinflammation, and there are several potential areas for use. I know the drug companies are very interested in this drug for MS, for example. I know that there's local interest in this drug in HIV populations and patients with depression, chronic depression, and studying different molecules potentially as therapeutic agents for these areas. And so this is from a study published in our medical journal, the Journal of Nuclear Medicine, which compares the F18 labeled version of this agent to the C11 labeled version, the PK11195. You can see they have similar binding characteristics to the target, but the benefit of the new drug, GE180, kind of over the C11 version is similar to that of carbon-11 PIB versus fluorine-18 PIB, in that it's an attractive target for a companion diagnostic, in that labeling the compound with fluorine-18 allows drug companies to have broader access for this drug. And the F18 label compound is also one that is accessible in an FDA GMP format, whereas there hasn't really been much work done with the C11 compound or molecule in the GMP area. So a good bit of work would need to be done there to make it up to GMP standards to support FDA clinical trials, whereas the F18 labeled agent is kind of already beginning to see use in clinical trials for therapeutic intervention in these various areas. So kind of some bullet points to leave you with. There's a lot of obstacles associated to making this technology available. Again, there are major academic institutions and universities that have little or no access to these molecules, that are kind of in a race to establish these programs. The regulatory hurdles are very high. It can take 4 to 5 years to establish a new lab. We're very lucky here to be close to the medical school in Johnson City and already established and approved to manufacture these agents. And the use of companion diagnostics and development drug discovery is growing rapidly. This field essentially didn't exist maybe 6 years ago, 8 years ago, as it is today, and now virtually every drug company is exploring the use of these companion diagnostic agents and development of drugs for therapeutic use. And it spans from the preclinical setting now to test portfolios of biomarkers for potential targeting use and development of a therapeutic agent, to biodistribution studies, clinical trial subject enrichment, selecting appropriate patients for a given clinical trial based on the absence or presence of the target. And then again, post-approval clinical management. Once these drugs are approved in a clinical setting, these companion diagnostic molecules would be kind of part and parcel with the clinical management and appropriate use for management of these patients. So, we believe that the use of companion diagnostics and drug development and research will continue to expand probably exponentially over the next several years. So, it's kind of a conclusion of the overview of kind of this evolving area. Does anyone have any questions? Dr. Kendall.
Thank you again. That was really informative and wonderful. I was reading through a paper relatively recently looking at the use of endotoxin to induce microglial activation. But in that paper, they were using PBR28.
Mm-hmm.
Are there advantages or disadvantages in PBR28 versus the G180 compound when looking at microglial activation?
Yeah, so I think, the answer to that question, there's two ways to answer that question. I think on a molecule-to-molecule basis, I think that the other molecule that you mentioned has some promising characteristics associated with it. The issue is these portfolio of drugs, it's kind of a path of least resistance once some of the preliminary toxicity pharmacology work has been done and some of the GMP work, legwork is being done. So I think kind of the drug that's most available today is the GE180 drug. More work has been done in that area to make that drug in a more GMP environment. But I can tell you that there is a lot of interest in this drug that a year ago there was no interest in this drug, and over the last 6 months the interest in this area has grown quite rapidly. So what we typically see is that there are fast followers whenever there is an important pathway and target identified, and so what I anticipate seeing is more work being done on that molecule and other molecules kind of in the same space by other drug companies. But currently, as of today, really the only one that's available, kind of a GMP backbone, is kind of the GE180 at this point. But I anticipate that we'll see kind of the either the emergence of the tracer that you mentioned and probably other competitive tracers as time goes on and work is done in this area with 180. There will be fast followers and improved, new and improved versions of that molecule down the road. So,
thank you very much.
Mm-hmm.
Actually, related to that point and HIV-associated neurocognitive disorder, the work that's been done so far using PK11195 has been mixed in
Right.
showing differences that are we would expect for microglial activation to be increased with patients with neurocognitive disorder rather than those with no neurocognitive impairment. So this would be an area of relevance too for second-generation tracers that might generate better signal
Yeah, good
for
point.
TSPO.
Yeah, good point. I mean, an example of this conversation with another tracer, I had this conversation Wednesday night with the drug company. So tau tracers that are currently available, there's off-target binding, right? And so the ideal tracer has a very high signal to background. So you want the tracer to be very specific for a given molecule or marker or target. And but also because we're trying to be very discrete and mimic other normal physiologic processes, we're always going to have off-target binding. So the discussion I had earlier this week, the tau tracers that are currently being used in research, one tau tracer also has off-label binding in amine oxidase A, and the other one has off-target binding in MON-B. So kind of the drug companies are both going back to kind of the drawing board to try to develop more and more specific molecules that target only the tau protein and not these other unintended off-target labeling of other molecules kind of in the same physiologic space. So there's constant work being done to make these molecules more and more specific. Now, could they be useful in a clinical setting already? Probably, but in drug development and discovery, the more specific they are because we're able to quantify them, the better off we are. So we'll continue to see evolution in not only these tau drugs, but probably also the TPSO labeling agents as well as time goes on. Any other questions? Okay. Thank you all very much.